Welcome back to Biotech Wonders, the captivating channel dedicated to biotech techniques. In today's video we will talk about SDS-PAGE, also described as vertical gel electrophoresis. Here is the outline of this video. We will discuss the function of SDS, the discontinuous gel system used in SDS-PAGE, the formation process of the polyacrylamide gel, and the underlying principle behind the separation of protein molecules using this technique. So friends, first let us understand what is SDS-PAGE. SDS stands for sodium dodecyl sulfate and PAGE stands for polyacrylamide gel electrophoresis. SDS is an anionic detergent with negative charge, used as a strong denaturant to break the protein structure. Proteins have both positive and negative charges due to the presence of amino and carboxyl groups. SDS along with beta-mercaptoethanol breaks the disulfide bonds and unfolds the protein into a linear structure with a coating of negative charge imparted by SDS molecules. This allows the protein to migrate toward the positive end of the gel and be separated. Now, let's understand the two gel systems involved in SDS-PAGE. Each gel has a unique purpose. The first is the stacking gel with a pH of 6.8 and 3 to 5% acrylamide, while the second is the resolving gel with a pH of 8.8 and 10 to 15% acrylamide. Let's now focus on the key ingredients used in these gels. Both gel is prepared using acrylamide, bisacrylamide, buffers with different pH levels, SDS, timid tetramethylethylenediamine diamine, and APS, ammonium persulfate. APS and timid are the two important components for the polymerization of acrylamide gel. APS spontaneously generates free radicals that are highly active, and these radicals are stabilized by timid. The stabilized free radicals, in turn, activate the acrylamide monomers, which subsequently combine with bisacrylamide molecules to produce polyacrylamide gel. The linear chain of acrylamide monomers is held together by hydrogen bonds, while bisacrylamide connects the two linear polyacrylamide chains. This is the SDS-PAGE gel containing upper stacking gel, pH 6.8, and lower resolving gel, pH 8.8. The gel is submerged in a buffer tank containing trisglycine buffer with a pH of 8.3. To initiate the gel run, it is connected to a power supply with the positive electrode at the bottom of the tank and the negative electrode at the top. Using a pipette, samples and a protein ladder are loaded into the wells of this gel. To visualize the sample run on the gel, the protein samples are premixed with bromophenol blue dye. The trisglycine buffer provides glycine and chloride ions, which helps to concentrate proteins of different sizes into a compact horizontal zone inside the stacking gel, so that they enter the resolving gel at exactly the same time. Inside the resolving gel, the proteins are separated based on their molecular weight. Here is the take-home message from today's video. SDS denatures the protein and provides negative charge to them. Beta-mercaptoethanol is a reducing agent which breaks disulfide bonds. This continuous gel system of SDS-PAGE consists of two gels, namely stacking gel, pH 6.8, and resolving gel, pH 8.8. TMED and APS are catalytic activators for polyacrylamide gel formation. SDS-PAGE separates proteins based on their molecular weight. Thanks for watching. Stay tuned for next upcoming video on how to perform SDS-PAGE.